Welcome you on the next my lectures. It's about application of phytohormones in increasing crop productivity in natural and stressful conditions. Over the next hour, I hope, of course, I will explore how phytohormones, this essential plant signaling and important to plant growth and development substances can be harnessed to improve crop yield, enhance resilience to environmental stresses, abiotic and biotic of course, and support global food security. With climate change and a rapid population growth placing immense pressure on agriculture understanding and manipulating these natural compounds is more and more important than ever. Agriculture today faces unprecedented challenges. I said about climate change, rapid urbanization, and shifting food consumption patterns are significantly affected global food security. Rising temperatures in the world, unpredictable rainfall, sea degradation, and increasing biotic stresses such as, for example, pests, threaten crop yield worldwide. Additionally, as the global population continues to grow, the demand for food production is reaching critical levels. To address these changes, scientists and agricultural experts are focusing on developing climate-smart crop varieties, crops that are not only high-yielding but also more resilient to environmental stresses. One of the most promising approaches in this pursuit is the application of phytohormones. We know that naturally occurring small organic molecules in plants act as signaling molecules and have a very important fundamental role in plant biology. By understanding how these hormones function and how they can be manipulated, we can enhance crop productivity and resilience of plants, of course. In modern agriculture and plant biology, improving crop yield, productivity and stress resilience is a top priority. Both biotic stresses such as pests and pathogens and abiotic stresses including drought, extreme temperature and soil salinity can lead to substantial crop losses worldwide. As the global population continues to expand, the demand in high yielding or more resilient crop is increasing. Meet to these challenges, scientists are exploring various strategies, including metabolic engineering of phytohormones. I presented a similar slide on the previous lectures, so I think that I should omit it. Similar like graph which represent the structures of main groups of phytohormones, which of course I presented earlier. Plants do not rely on a single hormone for survival. Instead, they use, we know, a complex network that work together to regulate growth, development, and responses to environmental changes. Phytohormones can have both positive and or negative interactions, allowing plants to finally tune their biological processes. To maintain optimal growth and stress adaptation, plants precisely control hormone levels through biosynthesis, degradation, and inactivation. This regulation ensures that the right hormone is present at the appropriate time and is right placed within the plant. Although phytohormone exists throughout the plant body, their concentration is very special and temporary. For example, hormones involved in growth and development, such as auxin, cytokinins, brasinosteroids, are often found in specific plant tissue where active growth is occurring. By understanding how these hormones are synthesized, transported, and degraded, scientists can develop strategies, special strategies to enhance plant growth and improve stress tolerance and increase, of course, agricultural productivity. Photosynthesis is the very important fundamental of plant growth and productivity, and instinctively all plant hormones play a role in either enhancing or regulating this process. By modulating photosynthesis, phytohormone directly impact biomass accumulation, stress resilience, or overcrop yield. Different phytohormones contribute to this regulation in various ways. For example, auxin, cytokinins, gibberellins, brasinosteroids are primarily involved in plant development, promoting plant cell expansion or growth organ, which can enhance photosynthetic capacity. In the next lecture, Professor Hayat presents the detailed regulation of photosynthesis by brasinosteroids. Because this distinct role of phytohormone engineering and quantification can offer a valuable insight into how different tissues function during various development stages and developmental conditions. As we can see, phytohormones play a critical role in development and stress responses. Plant hormone levels are influenced by two major processes. Anabolic pathways, that mean biosynthesis. Catabolic processes, that mean degradation and inactivation, where the plants break down or regulates hormone levels. Of course, both pathways are essential for maintaining hormonal balance. By targeting the genes and enzyme involved in these pathways, we can enhance traits such as increased drought tolerance, faster growth, and improved resistance to diseases. Beyond biosynthesis and degradation of plant hormones, another potential target for improving growth is hormone signal transduction pathways. By modifying the receptors, transcription factors, or regulatory proteins involved in hormone signalings, we can fine-tune plants' responses to environmental conditions. To manipulate phytohormone levels efficiently, scientists have identified specific target genes involved either hormone biosynthesis as presented as green or catabolic presented as red in red color. These genes can be modified through genetics engineering or selective breeding to enhance desirable traits in crops. Some key enzymes include, for example, gibberellin 20 oxygenase, gibberellin 3 oxygenase, and gibberellic 2 oxygenase, which regulate the gibberellic metabolism, affected plant height and growth. Another auxin biosynthesis gene, for example TMS1, which influence auxin production, critical for root and shoot development. Cytokinase SKX cytokinin oxidase dehydrogenase, which controls cytokinin degradation, impacting leaf longevity and stress resistance. Another genes, NCED and LOS5, which regulate abscisic acid biosynthesis, are crucial for stress tolerance. And etc., of course, ACC synthase, ACCI oxidase regulate ethylene production, which influence fruit ripening and stress responses. By selectivity over expressing or silencing these genes, we can successfully create stress resistance, high yielding plant varieties. For example, auxin are among the most important phytohormones, playing a crucial role in cell differentiation, vascular tissue development, formation, apical dominance, and tropic responses. How dominance we can see outside this building and the front of the Department of Botany, Al-Gah Muslim University. It's a very good example for this apical domination. By manipulating, of course, auxin biosynthesis and signaling, we have been able to enhance plant growth and productivity. One of the key auxin is indole-free acetic acid. IAA genetics engineering has allowed us to modify its biosynthesis and regulation. For example, of course, IAM gene from Pseudomonas syringa has been used to introduce parthenocarpy, leading to the seedless fruit development in crops like tomatoes and grapes, increasing productivity. The next gene, TMS1 from Agrobacterium tumefaciens, were expressed under tuber-specific promoters, has successfully enhanced tuber formation in potatoes, contributing to higher yields. The next key genetics modification include overexpression, the Arabidopsis UK6 gene in potatoes, and the similar genes in poplar resulted in increased drought tolerance and erect plant structure, and reduced, of course, water loss, making these plants, that mean, for example, potatoes or polar, better suitable for arid environments. The next gene, GH3 gene family responsible for auxin homeostasis, play a significant role balancing auxin levels in plants. Overexpression Oryza sativa GH3.1 in rice and enhance resistance to pathogen. Similar genes from this family Oryza sativa improved cold tolerance by reducing free auxin levels, preventing excessive growth under stress. These examples highlight how fine-tuning auxin levels can help crops adapt to environmental challenges while maintaining productivity. By controlling not only, but in this case, auxin levels, we can optimize root and shoot development, improve nutrient updates, and increase plant resilience to environmental stressors. Beyond auxin biosynthesis and homeostasis, we know that auxin move within the plants, tightly regulated to ensure proper development and stress adaptation. Overexpression on AVP1, vascular H plus pyrophosphatase, enhances auxin transport, leading to increased cell division and overall plant growth. This modification has been particularly used in improving root architecture and enabling plants to absorb water and nutrition more efficiency. In addition to transport, auxin signaling pathways present under opportunity for crop improvement. That means that auxin signaling mechanism involves key elements such as TRR1, AFB, co-receptor, AUX, IAA transcriptional receptors, and ARF transcriptional factors. These elements can regulate how plants perceive and respond to auxin, making them potential target for transgenic modification to enhance stress tolerance and, of course, yield stability. Furthermore, auxin mutants offer insights into stress adaptive traits. For example, UC71D mutants exhibit auxin overproduction, leading to enhanced drought tolerance and altered plant morphology. The next mutants shown in rice linked to auxin hydrolyse activity have resulted in increased grain weight and improved crop yield. Cytokinins are another essential phytohormones which play a critical role in differentiation, leaf senescence, nutrient allocation, and other physiological processes. By manipulating cytokinin levels, we can significantly improve plant growth, productivity, and of course resistance to environmental stressors. Key genes involved in cytokinin metabolism have been targeted for genetics manipulation. If, for example, is IPT, isopentenyl transferase, is a crucial enzyme in cytokinin biosynthesis, responsible for promoting cell division and delaying senescence. The next, CKX, on which I provide many studies, cytokinin oxidase dehydrogenase, regulate cytokinin degradation, controlling plant growth and development. UGT catalyzes cytokinin conjugation, affecting its activity and stability in plants. By enhancing cytokinin biosynthesis or reducing its degradation, we can develop crop with delaying aging, improved drought resistance, especially SKX, and higher yields. While cytokinin offer numerous benefits for plant growth, excessive cytokinin production can sometimes lead to unintended effects such as uncontrolled growth. To overcome this challenge, researchers have developed strategies for conditional expression, activating cytokinin biosynthesis only under stress, such as drought of aging. There are two key approaches, and they have been successfully tested. It's PIT expression under the promoters SAG12, which using the senescence associated with SAG12 promoter to drive IPT expression, allows plants to produce auxin only during leaf aging. It leads to delay senescence, prolonged photosynthesis, and improved biomass accumulation. As seen in tobacco and creeping bent grass, it's similar, its expression IPT under SARC promoter. The SARC promoter is stress inducible, meaning IPT is activated only under drought conditions. It has been tested in rice, in maize, and peanuts, where it significantly improved water use efficiency, drought tolerance, and the crop yield under water deficient conditions. This advancement showed that by precisely controlling cytokinin expression, we can enhance plant productivity, stress resistance without compromising overall growth and development. One of the most valuable applications of cytokinin manipulation in agriculture is its role in delaying leaf senescence, which directly affects photosynthesis, biomass accumulation, and crop yield. We know that cytokinin naturally prevent premature aging by maintaining chlorophyll contents and sustaining photosynthetic activity. Through genetics engineering, scientists have successfully extended the production lifespan of plants. For example, IPT expression under SAG promoter has been shown to delay leaf senescence, leading to the higher biomass improved yield. And the next, of course, which I said earlier, reducing SKX affect results in elevated cytokinin levels. Beyond cytokinin biosynthesis and degradation, another crucial area for improving crop performance is cytokinin signaling. This pathway determines how plants perceive and respond to cytokinin, influencing growth, development, and stress adaptation. In cytokinin signaling pathway consists of three major components: histidine kinase receptor, AHK, and these proteins detect cytokinin and initiate the signaling cascades. The second is histidine transfer proteins, SHP, and these act as messenger, transferring signals from receptor to response regulators. And another, the third, the response regulators, ARR, the type B especially activates genes that enhance growth and stress response, while type A, RRAs, function as negative regulators, fine-tuning the process. By manipulation these signaling components, we can, the first, enhance cytokinin sensitivity, leading to the better growth regulation and yield stability. And the second, modify stress response signaling, improving plant adaptation to drought, salinity, and nutrient limitation. And the third, balance cytokinin interaction with other phytohormones, can optimize developmental processes without unwanted side effects. Studies on cytokinin manipulation has provided promising results. in improving plant productivity and stress resilience across various crop species. Here are some key findings. I said about IPT expression in rice and the maize. I said also on SKX silencing barley, which reduces cytokine degradation, has led to higher cytokine retention, and of course improved growth and increased yield in plants. And overexpression other gene, for example in tobacco, has improved salt stress tolerance, demonstrating cytokines' role in enhancing abiotic stress resistance. These experimental outcomes confirm that cytokine engineering is a valuable strategy for developing high-yielding and stress resilience crop. The next, I would like to go to another group, gibberellins, which by manipulating their levels, we have successfully enhanced plant growth, also biomass production and stress resilience. For example, these genes involved is one-in-one final step of biosynthesis, gibberellin 20-ox, increasing gibberellin levels and promoting growth and elongation. The next, of course, enhances active gibberellin production, is crucial for seed germination and flowering. The next, GA2 oxidase, is responsible for GA degradation, reducing growth and leading the dwarf phenotypes, which are beneficial for center crop traits. By controlling the expressions of this gene, we can fine-tune plant height, growth rate, and stress adaptation. This, of course, application demonstrates that gibberellins engineering offers great potential for optimizing plant, not only architecture, but improve yield and enhance stress tolerance. Maybe the same. This is another example, use signal transduction. Maybe I would like to go to another hormone. Abscisic acid is often referred to stress hormone because it plays a central role in plant response to abiotic stresses, particularly drought, salinity, and cold stress. Abscisic acid is responsible for regulating several key physiological processes, including the first, stomatal closure, seed dormancy, embryo morphogenesis, and synthesis of protective protein and osmoprotectants. Some that mean that abscisic acid signals guard cell to close stomata, in stomatal, of course, closure, reducing water loss under drought conditions. In the second case, abscisic acid prevents premature germination, ensuring seeds only sprout when environmental conditions are favorable. In the next case, for example, synthesis of protection protein, this component help plants withstand dehydration and oxidative stress. Since abscisic acid is directly linked to drought survival, manipulating its biosynthesis and signaling pathways present a valuable strategy for enhancing crop resilience to water-deficient environments. To enhance drought resistance and stress adaptation, the key genes involved in ABA biosynthesis are ZIP, zeaxanthin epoxidase, NCED, that's mean 9-cis-epox carotenoid dioxygenase, and LOS5 ABA3 encodes molybdenum cofactor, which is essential for activation abscisic acid aldehyde oxidase, which is in the final step of biosynthesis of this hormone. Of course, by increasing this expression of these genes, we can successfully develop a crop with higher abscisic acid levels, which results in enhanced drought tolerance, better water use efficiency, and improved survival under extreme conditions, which have in natural in addition to improving drought and salt tolerance, abscisic acid engineering has also been shown to enhance plant recovery after stress exposure. When plants experience extreme conditions such as, for example, drought, the ability to bounce back after rehydration is crucial for maintaining productivity. I show some key findings in this area. Includes petunia plants overexpressing tomato, and SED1 showed complete recovery after 4 days of drought once rehydrated. It's demonstrate ABA, abscisic acid role in post-stress adaptation. Several, of course, findings, strategies, plants with enhanced ABA signaling displayed faster reactions of growth processes after drought stress, ensuring better yield stability. These studies suggest at present that engineering abscisic acid pathways can improve not only resistance to stress, but also the ability of crops to recover quickly, reducing yield losses after adverse environmental conditions. Maybe the same. Oh, I thought about it. Ethylene. Ethylene is a small gaseous, very molecules, but very important. Ethylene influencing several critical physiological processes, including flowering senescence and leaf abscission. Ethylene regulates aging processes, helping plants shed leaves and flower when they are no longer beneficial. And the second, of course, the fruit ripening. It's accelerate the ripening of climacteric fruits such as banana, tomatoes, and apples, making it an essential hormone for agricultural and food industries. Response to biotic and abiotic stress. Ethylene is involved in stress signaling, helping plant adapt to drought, flooding, head, cold, and pathogen attacks. Because of its dual role in both growth promotion and stress responses, ethylene manipulation presents both opportunities and challenges for crop improvement. To optimize ethylene's effects on plant growth and stress adaptation, we can focus it on modify its biosynthesis and signaling pathways. Ethylene biosynthesis is controlled by two key enzymes. ACC synthase is aminocyclopropano-1 carboxylic acid synthase. This is an enzyme catalyzes the first step in ethylene production and is a rate-limiting factor in ethylene biosynthesis. The next is ACC oxidase, convert ACC into ethylene, determining how much of this hormone is available in the plant. In addition, the ethylene signaling pathway includes ethylene receptors. It's ETR1, 2, ERS1, Per2, EIM4. This protein detects ethylene and initiate absence of this hormone. Active genes involved in stress response, growth regulation is EIN3, EIL transcriptional factors. And the next, of course, ethylene response factors, which mediate ethylene effects on gene expression. By targeting these pathways, we can, the first three, reduce ethylene production to delay fruit ripening and extend shelf life. And second, enhance ethylene signaling to improve stress response, making plants more resistant to drought, salinity, and pathogens. And third, modify ethylene sensitivity to balance growth and defense responses. Ethylene engineering has had a significant impact on fruit production, offering, of course, valuable solution for modern agriculture. The key application includes, of course, reducing ethylene production for delay ripening. For example, transcription melons with antisense, ACC oxygenase exhibit slower ripening, improve texture and higher sugar contents, extended the market shelf life. Apples and tomatoes with suppressed ethylene biosynthesis have firmer fruit and better storage potential, reducing food waste. It's very, of course, important too. And the second, enhancing ethylene senescence for stress tolerance. For example, ethylene insensitive maize mutants demonstrate higher grain yield under drought conditions, highlighting its role in improving agricultural resilience. The next show mutants, ACS7 mutants in Arabidopsis, of course, is not a crop plant, showed increased model of plant in biochemistry of molecular sciences. It showed increased tolerance to salt, osmotic, and head stress. Another key is microbial application in ethylene regulation. This means rhizobacteria producing ACC, deaminase, break down ethylene precursors, which reduce ethylene levels in plants and improve drought resistance. Summarizing, these findings demonstrate that controlling ethylene biosynthesis and signaling can optimize both food production and stress resilience, making agriculture more suitable. In the next slide, I would like to present challenges and considerations. While ethylene manipulation provides numerous benefits, including improved stress tolerance, enhanced fruit storage, and optimized plant growth, it also presents significant challenges that must be carefully addressed. One of the key issues is ethylene dual role in plant development and stress responses. While reducing ethylene production can delay ripening and improve growth tolerance, excessive suppression may negatively impact normal growth process and weaken the plant's stress resistance against pathogens. On the other hand, increasing ethylene levels can enhance stress resilience, but it may also lead to premature aging and reduced crop yield. To ensure optimal outcomes, precise regulation of ethylene-related genes is, of course, essential. Instead of completely blocking ethylene production, targeted genetics modification using conditional promoters allow ethylene expression only under specific environmental conditions. Another promising strategy is to modify ethylene receptors rather than biosynthesis genes, providing more controlled responses without disturbing overall growth. We know about that brazinosteroids are polyhydroxysteroids, influence cell elongation, cell division, and making them, of course, essential for normal plant physiology. One of the most important roles of brazinosteroids is in cell elongation and division, which contribute to overall plant height, leaf expansion, and root development. They also regulate processes such as embryogenesis, pollen tube growth, and senescence, ensuring that plants undergo proper reproductive development. Brazinosteroids have been found to enhance photosynthesis efficiency by improving chloroplast function and increasing carbon assimilation, ultimately leading to higher biomass production. In addition to their role in normal, of course, in normal conditions, brazinosteroids help plants withstand environmental stress. Under drought conditions, brazinosteroids improve water use efficiency by regulating stomatal conductance. In saline environments, they enhance ion homeostasis, reducing the toxic effect of excess sodium accumulation. In another example, in response to heat and cold stress, they activate protective mechanisms that help maintain cellular integrity. These properties make, of course, brazinosteroids an attractive target to gene manipulation and agricultural application aimed at improving crop resilience and yield stability. To enhance plant growth and stress resilience, we can focus it on modification, biosynthesis, and signaling pathways of brazinosteroids. These hormones are synthesized through serious enzymatic reactions with key genes controlling the production and activity. One of the most, of course, important genes is L04 dwarf. Thank you. L04, which encodes steroids 24 alpha hydroxylase, a key enzyme in brasinosteroids biosynthesis pathways, which I will show in the next lecture, entitled around the brasinosteroids in algae. Of course, the biosynthesis pathways I show. Overexpression of this gene has been shown to increase plant size, improve yield, and enhance stress tolerance in multiple crop species. In addition to biosynthesis, the brasinosteroid signaling pathways plays a crucial role in determining plant responses to environmental conditions. We know that receptor in this transduction is BRI1, BAC1 receptor complex is at the center of brasinosteroid perception, triggering downstream signaling cascades that regulate gene expression related to growth and stress adaptation. Further downstream, signaling components such as BSK13, BN2 kinase, and transcriptional factors like BZR1 mediate brasinosteroid responses, balancing cell expansion and stress tolerance. Brasinosteroids have been widely studied. Thank you again. Widely studied for their ability to enhance plant growth, productivity, and stress tolerance. By optimizing their biosynthesis and signaling, we have successfully improved. We can successfully improved crop performance in various agricultural settings. One of the most significant applications of brasinosteroids is in increasing grain and biomass yield. Studies have shown that overexpression of L04 gene in rice led to 15 to 44% increase in grain yield, while in Arabidopsis as a model plants, the same modification results in larger plants and 59% increase in seed yield. Of course, it's not important. Comparison is maybe enough. It's not necessary. In addition to improving growth, brasinosteroids play a crucial role in helping plants cope with environmental stresses. About heavy metals, I will talk later. Exogenous application of brasinosteroids such as 24 AP brasinolide or 24 homo brasinolide has been found to enhance photosynthetic efficiency and stringent antioxidant defense system, particularly in tomato and eggplant under heat and salinity stress. By the way, please see the Professor Shamsul Hayat publications in detail, which show. Brasinosteroids have also been shown to mitigate the toxic effects of heavy metals, as said, such as, for example, cadmium in tomato, making them available tools in improving plants' health in polluted soil. Brasinosteroids, foliar spray in cadmium-stressed tomato plants, efficiently neutralize heavy metal toxicity and increase fruit yield, proving that this hormone can help plant thrive in a polluted soil. Similar in rice and the overexpression of exordium, exogene, a brasinosterol responsive regulator, resulted a large leaf and root system, leading to improved nutrient uptake and high biomass production. Another breakout involved application of erect leaf rice mutants with altered brasinosteroid signaling, which demonstrate greater light penetration into the, for example, canopy, increasing photosynthesis and boosting grain yield. And this finding shown how brasinosteroids can be manipulated not only to enhance plant growth, but also to improve overall resource use efficiency, making crops more productive and resilient. And these studies confirm that brasinosteroid engineering is a promising approach in modern agriculture. While brasinosteroid enhance plant growth and stress resilience, their application must be carefully controlled. Additionally, brasinosteroids interact with other hormones, for example, auxin and cytokines, meaning that enhanced growth may come at the cost of reduced stress tolerance. To address this challenge, we are exploring target genes modified that regulate biosynthesis, regulate brasinosteroid levels in specific tissue and or developmental stages. Additionally, combining brasinosteroid engineering with other phytohormonal modification, such as gibberellins, for high control or abscisic acid for drought tolerance, can help achieve a more balanced and optimized crop phenotype. By carefully fine-tuning brasinosteroid biosynthesis signaling and interactions with other phytohormones, we can develop high-yield stress resilience crop with maintain a strong structural integrity. The next group which I would like to discuss is yasmonates. Yasmonic acid is a crucial group, is crucial hormone involved in plant defensive stress adaptation and development. They play a central role in responding to wooding, herbivore pathogen attacks, and other environmental stresses such as salinity, drought, and cold. Beyond the role in defensive, yasmonates also regulate seed germination, root growth, tree formation, flowering, and senescence. This hormone act as signaling molecules that activate protective responses, allowing plant to adapt to challenges and environmental. Due to their role in stress resistance and growth regulation, yasmonates have become a key focus in agricultural biotechnology. By modifying yasmonate pathways, researchers aim to develop a crop with stronger pest resistance, enhanced drought tolerance, and improved overall resilience. To enhance this plant defense, we have focused on modified yasmonate biosynthesis and signaling pathways. Of course, I present the key enzyme in yasmonate production includes 13-lipooxygenase, 13-LOX, which catalyze the first step in yasmonic acid synthesis, and yasmonic acid carboxyl methyl transferase, JMT, which converts yasmonic acid into its active form, as mean methyl yasmonate. In addition to biosynthesis, yasmonate signaling pathways involves CO1 receptors, JAZ receptors, MYC2 transcription factors, which regulate plant response to stress. Of course, the genetic modification targeting these compounds have led to improved resistance to pathogens, enhanced drought tolerance, and increased yield stability. By fine-tuning yasmonate biosynthesis and signaling, we can develop crops that are more resistant to pests and environmental stresses while maintaining optimal growth. In the next slide, I would like to show this modification. Maybe this is similar what I said. Yasmonates, maybe this is not you're just interested. Something else. Phytohormones, of course, play a critical role in protected plants against biotic stresses. One direct defense strategy is the production of secondary metabolites such as alkaloids, cyanogenic glycosides, and glycosinolates. This compound act as a natural pesticide, deterring herbivores and inhibiting pathogen growth. Phytohormones such as yasmonate and yasmonic acid regulate the biosynthesis of these defensive compounds, ensuring to release the volatile organic compounds which attack natural enemies of insect pests. The signaling molecule, that's mean yasmonic acid and salicylic acid, guide predatory insect to the prey and enhancing biological pest control. Key volatiles such as farnesan, methyl salicylate, and oxymen have been identified as a major player in this plant-insect communication network. The future of phytohormone research lies in integrating advanced technology to further enhance plant productivity, stress resilience, and sustainability in agriculture. One of the most promising area is plant hormonomics, which combines omics technologies to analyze hormone interaction at the molecular level. It allows scientists to gain deeper insight into how phytohormones regulate growth and stress responses. Another key focus is the development of climate resilience crop through target hormonal manipulation. With climate change posing increasing challenges to food security, so research are engineering crops that can better withstand extreme weather conditions, including drought, heat, and soil salinity. By fine-tuning phytohormone pathways, we can improve water use efficiency, nutrient uptake, and resistance to water environmental conditions. Enhancement in biosensor and omics tool are also revolutionizing phytohormone research. These technologies enable precise hormone profiling, allowing for real-time monitoring of hormone level in plants. It's of course future. It can lead to more effective breeding problems and agricultural practice that optimize hormone regulation for maximum yield and resilience. Finally, Sustainable agricultural strategies are increasingly leveraging hormone-based approaches to reduce resilience on chemical fertilizers and pesticides. By harnessing the natural signaling power of phytohormones, future crop management practice will become more environmentally friendly and efficient. What's the next? Phytohormone as a play a fundamental role in regulating and stress, the ability to enhance crop productivity and resilience makes them invaluable tool for modern agriculture. A comprehensive understanding of phytohormone mechanism will allow research to optimize their application, balancing growth, defense, and adaptation. The integration of genetic engineering, precision agriculture, and omics technology will further expand the potential of phytohormones in creating more effective and sustainable future for agriculture. So thank you very much for your attention.